Hello everyone, welcome back to the class. In our previous class, we studied about transposable elements in prokaryotic organisms and eukaryotic organisms. The transposable elements which are present in the prokaryotic organism are known as prokaryotic transposon. Example for that is IS elements. The transposable elements or the transposons present in eukaryotic organisms are known as eukaryotic transposons. Example for that are retrotransposon, that is LTR and non-LTR, P elements in Drosophila, controlling elements in maize. The bacterial transposon is a type 2 transposon where it transposes directly to the DNA and it follows the particular mechanism, that is cut and paste type of mechanism, where the transposable element, that particular sequence is cut in the donor DNA and it is pasted into the recipient DNA or the target site. They produce short direct repeat at site, and we now know that how these short direct repeats are formed. It is during the transposition of the particular transposon in the bacterial genome. And example for bacterial transposon is IS element. It is having the inverted repeats which are in green color in this image, and they are having a protein coding region. The region between the two inverted repeat is known as protein coding region, where it codes for a particular enzyme, that is transposase, and they have short direct repeats that are in pink color in this image. So these are the flanking direct repeats. So let us revise this also, that is transposition of IS element. First, the staggered cut is made. The staggered cut is like this, and it is having uneven ends where there is a transposable elements. The transposable elements is inserted, and you can see here the void, the gap. That gap is filled by the enzyme, that is DNA polymerase, and that will form a flanking direct repeats. Next, we learnt about composite transposon, which is very composite in its structure, and in addition to IS elements, bacteria contain composite transposons where they contain one or more protein coding genes. In here, the TN10 is a typical example for composite transposon, which is having tetracycline resistance gene in addition to the transposase gene. And we also studied about the eukaryotic transposon, especially in P elements, that is found in Drosophila. It is 2907 base pair long, and they have inverted repeats, and they have here long terminal repeats, that is LTR. In eukaryotic transposon, especially it is a retrotransposon, and in retrotransposon we know that it is LTR and non-LTR. The transposable elements having long terminal repeat is known as LTR. The transposable elements which lack these LTR regions are known as non-LTRs. So they do not utilize any RNA intermediate during transposition. They can insert at many different positions in the Drosophila chromosome. And the controlling elements in maize is having the inverted repeats, the flanking direct repeats region, and they have two classes of controlling elements, that is dissociation and activator. DS is lacking the transposase gene. AC is having all the apparatus needed for their transposition. So this DS will take help from this AC in order to get transposed. So AC is known as autonomous transposon, as they can move and they can synthesize their own transposase enzyme. DS is non-autonomous, they can't move as they don't have transposase gene. And retrotransposon here, which is LTR, is directly or it is elaborately explained here. LTR retrotransposon is found in viral transposon. It is similar to retroviral DNA. They are flanked by 250 to 660 base pair LTR. They encode reverse transcriptase and integrase. They move in genome by being transcribed into RNA, which then undergoes reverse transcription, and they are found in yeast, TY elements. They, as I discussed this concept or this particular topic in Drosophila, that is Copia elements. Non-LTR, that is non-viral retrotransposon, they lack LTR, that is long terminal repeats. They have AT rich stretch at one end. In invertebrates, it is signs, that is short interspersed nuclear elements, and lines, that is long interspersed nuclear elements. Viral transposon, we discussed about E. coli bacteriophage, that is MU, which is unusual phage. It is a temperate phage. It acts as both virus and transposon. Virus in the sense it affects or infects the bacteria. Transposon in the sense it transfers or transposes its element into the host DNA. So therefore it has both virus and transposon property. When DNA of MU enters the E. coli, its host, it integrates into a host genome. It integrates randomly into the host genome by the transpositional mechanism. So that's why it is having both type of property. DNA of MU is never found replicating as an independent molecule. So it needs some host. So it is not free of the chromosome, and the segments of the DNA packages into the virus particle contain whole MU genome plus small stretch of host DNA at the ends. So thus whole MU genome is a transposon that happens to have genes that package its DNA as well as genes that regulates its transposition. So till this we had discussed in our last class. So now today we will be learning about structure of eukaryotic chromosome. So there are many aspects to study here. So eukaryotes, if you think about a eukaryotic cell, you will always think about the nucleus as the central part, and it is a central core part of the cell where it acts as a monitoring compartment where it monitors the metabolism, functioning of the cell. So where you can, inside the nucleus, you can see the nuclear matrix, and as discussed earlier, there is a chromatin network which are of thread like structures like this, right? They are having thread like structures. And do you know what do we call this as? That is chromatin network. But what is the difference between chromatin and a chromosome? So chromatin and a chromosome, so these two terms you will be knowing, or you will be listening to this throughout this chapter, that is chromatin and chromosome. Chromatin and chromosome. See, this chromatin is a organized structure. It is a organized structure. What about chromosome? Chromosome is a highly condensed. It is highly condensed form. Both are DNA. Chromatin is less condensed. Try to differentiate both of this. So chromatin is also DNA, chromosome is also DNA. Chromatin is less condensed, chromosome is highly condensed. So chromatin is a organized structure. So I'll write it in this way: chromatin, chromosome. So chromatin is organized structure and it is made up of DNA plus protein. Where are these found? These are found in, oh, this is looking like F, right? I'll correct it. Now it is P. So DNA plus protein, and they are found in the nucleus, especially the G0 phase of nucleus. In G0 phase, the chromatin contains single double-stranded DNA. But this chromosome is highly condensed. And I have not mentioned here, it is less condensed. So what about this chromosome? Chromosome is made up of chromatin plus something new, that is nuclear matrix. And where it is present? It is present in the nucleus itself. It is highly condensed. Right? So chromatin is made up of what? DNA and protein. Chromosome is made up of what? Chromatin and nuclear matrix. Where it is present? Present in the nucleus. Where this chromosome is present? In the nucleus. How about chromatin's condensation? It is less condensed. How about chromosome's condensation? It is highly condensed. So let's see this structure. See here, genes are carried on chromosomes. See, it is highly compact, packed DNA, right? Here, it is extended DNA, packed DNA, extended DNA, chromosome, chromatin. So, chromatin is organized structure of DNA and protein. So, here you can see which protein we are talking about, histone protein. DNA plus histone protein makes chromatin, which is found in nucleus of G0 phase. Chromatin is less condensed. It is a extended DNA. Chromosome is highly condensed, packed DNA. Chromo in the chromosome word means color. Zome means body. It is strongly stained by particular dyes. In interphase nuclei, we can see chromatin plus nuclear matrix, which is a proteinaceous structure. DNA sequence attached to nuclear matrix is the matrix attachment region, that is MAR. Next, chromatin is further divided into euchromatin and heterochromatin. So, chromatin, let us summarize what we learnt about chromatin. Chromatin is less condensed. It is made up of DNA plus histone proteins. Now we know that protein is histone protein, and it is organized and it is having extended DNA. Extended DNA means loose DNA, like a thread. So I'll draw a DNA, extended DNA like this. Okay, there are some portions which are dark, and there are some portions which are light. Okay, so the light stained, less condensed portions are called euchromatin. The portions which are darkly stained are known as heterochromatin. Euchromatin and heterochromatin. So the light stained part are known as lightly stained, less condensed portion are known as euchromatin. The dark portion of the extended chromatin is known as heterochromatin. Let's now see light staining, less condensed, they are transcriptionally active. So light is equal to active. This is for your better understanding and better memorization. So light is active. They contain most protein coding genes. Active is having protein coding genes. Right, have an open accessible, as it is extended chromatin, it is open accessible chromatin structure. So if it is extended like this, methylation can happen, acetylation can happen easily. So that particular region where acetylation or methylation is having, that is mentioned as H3K4, H3K9. You will be learning about this in further classes. So for now, euchromatin is light stained, less condensed portion. They are active, transcriptionally active. They contain protein coding genes, and they have open accessible chromatin where methylation and acetylations happen. So now let's see about heterochromatin. Heterochromatin is dark, therefore they are transcriptionally silent. They remain condensed. But in euchromatin or in heterochromatin, there are two subtypes, that is constitutive and facultative. You will be learning about that shortly. Constitutive and facultative, they are distinguished based on the condensation state. For a particular time, the heterochromatin will become lightly stained, or it is converted to heterochromatin structure. Okay, that will be learned shortly. So heterochromatin is dark stained, highly condensed, and they remain condensed. They are transcriptionally silent. So further, constitutive and facultative. Constitutive means they remain condensed throughout the cell cycle and development. Remain condensed. They contain highly repetitive sequence. They are found at the pericentromeric, telomeric and loci along the chromosome. Though they are transcriptionally inactive, they replicate late, and they are reduced frequency of recombination. They are having reduced frequency of recombination. Facultative, they can change the regions of euchromatin converted to heterochromatin state. So it doesn't remain heterochromatin all the time. They can become euchromatin also. They have this potential for gene expression at some point in development. Either can be condensed or decondensed. So facultative, based on the faculty around it, it changes per time. So heterochromatin are of two types, constitutive and facultative. Constitutive remains always condensed. In the sense they are darkly stained, and they are dark or highly condensed, and they are transcriptionally inactive. Dark means inactive, light means active. So they replicate late, and they are having reduced frequency of recombination. Exact opposite to constitutive is facultative. Based on its faculty or based on its benefit, so it can be converted into euchromatin sometimes, or the regions of euchromatin can become heterochromatin based on this facultative, and has potential for gene expression. As it is sometimes euchromatin, so it has the property of transcription, and they can express the genes which are having, and either they can be condensed or decondensed. So this was about the chromatin types. See, chromatin DNA molecule has three specific nucleotide sequence that are nucleotide sequence are specific. They are centromere, telomere, and origin of replication. So this image shows the not all three, two regions you can see. The telomere, which is at the bottom, which is at the end, proximity end. Here centromere, it is at the center, and there is ori region, that is origin of replication, from where the DNA replication started. It is a region. All these regions is having a nucleotide which is specific in their sequence. So here in this you can see two types, or two major nucleotide sequence, or the chromatin DNA molecule having two specific nucleotide sequence. Let us discuss about centromere first. Centromere, as the name suggests, it is present at the center. What does it do? It is a constricted region. See here, it is a constricted region of eukaryotic chromatin. What does it do? There is a particular material or the element in the centromere that is known as kinetochore, which is assembled. In the previous image, you can see here, kinetochore are in green color, which is which is in close proximity to the centromere. So what does it do? In future, during the cell division, it pulls along with the spindle fiber, it pulls the chromatin or the sister chromatids to opposite poles in order to aid the cell division. So it is having a kinetochore and the sister chromatids are held together. So these are sister chromatids, 1, 2, 3, 4. 4 arms is known as sister chromatids. It is not located exactly in the center. Based on the position of that centromere, the chromosomes are divided into 4 types: metacentric, submetacentric, acrocentric and telocentric. Regions on either side of centromere, that is chromosomes arm. Kinetochore is associated with the centromere. It is a complex of protein. Kinetochore is what? Complex of protein, where spindle fibers attached to chromosomes during mitosis or meiosis help in proper segregation of sister chromatids. That is what I said now. So it helps in separating out, in dividing out equal number of chromosomes or the sister chromatids to daughter cells. There is no defined DNA sequence. It consists of large arrays of randomly repeated DNA sequence. So metacentric at the center, submetacentric just exact. It is not at the center. It is partially at the center. So it is submetacentric. Acrocentric is at the end. Telocentric, where full at the end or the apex region you can see the centromere. In interphase stage, chromatin replicates formation of 2 copies of each chromatin. In cells M phase, there is condensation of chromatin, formation of metaphase chromosome consisting of 2 identical sister chromatids. Held together at the centromere, the centromere part, it holds or it consists of 2 identical sister chromatids. Okay. Held together at the centromere, seen as constricted chromosome region. So there is a protein called cohesin, where this protein links sister chromatids and the centromere. Which protein? That is cohesin. Now let us learn about telomere. So telomere is a specialized structure. It caps the ends of the eukaryotic chromosome and it maintains the structural integrity. See, imagine a chromosome without a telomere, so that the DNA will be extended, the strand will be extended. So telomere acts as a boundary line. So it caps the end of the eukaryotic chromosome and it prevents the missing out of DNA sequences. So it ensures the complete replication of extreme ends of chromosome. So there is a significant importance of telomere in DNA replication. So it consists of long array of short and tandemly repeated sequence. It is of 100 to 1000 repeats. Unusual property of telomeric sequence is that it is having a G-rich single strand 3' overhang. That is a unusual property. G-rich, guanine rich single strand 3' overhang. So G-rich sequence is generated because there is limited degradation of C-rich complementary strand. This sequence of telomere has been highly conserved in evolution. So two sequence specific specific DNA binding proteins and telomere repeat, that is telomeric repeat binding factor TRF1 and telomeric repeat binding factor 2 TRF2, both of this bind directly with the telomeric sequence, which in turn interact with the larger protein. So the stability of chromosome ends is due to this telomere. Due to novel structure, T loop is found at ends of the mammalian telomere. Loops form at the ends of the chromosome under the direction of the protein TRF, that is telomeric repeat binding factor 2, which causes the 3' end of the chromosome to loop around the interdigitate into double helix, forming a loop. So here you can see the image of telomere. Next comes origin of replication. So you will learn about this much in detail during DNA replication class. So here we just discussing about the nucleotide sequence. In context of nucleotide sequence, origin of replication is chromatins are multi-repliconic. See, multi-repliconic means in the state that they have different or multiple regions of origin of replication, from where the replication initiates. So they are having more than one replicon, a replication unit. The individual replicons are relatively small, that is 40 to 100 kb. So each replicon has its own origin of replications. Okay, so see here, portion of eukaryotic chromosome is a multi-repliconic. It is having origin of replication here, 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 and here. So there are multiple regions of replication process happening or initiating. One chromatin contains multiple ori or origin of replication. In humans, chromatins have about 30,000 origin of replication, with each chromatin containing several hundred. So this is also made up of relatively small sequence, but they have particular nucleotide sequence. So this is enough for today's class. In next class we'll be learning about chromosomal organization. In this you will be learning about packaging of DNA into chromosome, how that loose chromatin threads will form chromosome, which is a completely intact packed structure, and how this packaging of DNA is initiated, who are helping them. So all that factors or all that aspects of chromosome organization you will be learning in the next class. Thank you.